Hello everyone, I'm Ronja from the Maastricht iGEM team. Welcome back to a new Geneducation video. Today we want to introduce you to ProteinProtein Interactions and a team from Berhampur that tries to solve a problem that results from unfortunate protein-protein interactions. Let's get started. Interactions between molecules play a fundamental role in all biological processes in our body. When molecules interact, they can create new compounds or work together in the body to keep us alive. In the so-called omics fields, such as genomics, which is the study of all genes and their interactions with each other and the environment, transcriptomics, which is the study of how genes are translated, or proteomics, which is the study of proteins and their interactions. Recent biological advances have enabled scientists to dig into the mechanisms of several cellular processes that keep us alive. At a molecular level, the major workhorses of our body, proteins, are some of the most crucial players in these interactions. Proteins build, maintain, and replace the tissues in your body. Our muscles, our organs, and our immune system are made up of mostly proteins. Proteins carry out their roles by interacting with other molecules, including DNA, RNA, other proteins, and other small molecules. Easy examples are muscle contraction, where actin and myosin interact with each other to make our muscles move. Or cellular transport, when a molecule docks into a tunnel protein to open it up and to let another molecule pass through the membrane into the cell. Protein-protein interactions, or PPIs, refer to the intentional physical contact between two or more proteins as a result of a biochemical reaction in the body. The interactions of proteins with each other creates a network of connected proteins and processes that keep us alive. Unfortunately, sometimes proteins interact with each other in a wrong way, and that leads to diseases. Therefore, efforts have been invested by scientists to try and find ways to block these interactions. One way to do that is by preventing the proteins from physically connecting to each other. By, for example, adding a molecule that is attached to the spot where the two proteins would normally connect. Then the proteins cannot connect anymore and therefore damage the body.
The iGEM team from IISER Berhampur is working on the dengue disease. Why is dengue a problem for the world? The virus that causes dengue, transmitted by the bite of infected Aedes mosquitoes, is very clever. It uses several complex interactions to modulate the immune responses of our body and divides and spreads throughout our tissues. PPIs are an integral part of these. However, because of the complicated pathways involved, there are several knowledge gaps in our understanding of the disease. Unfortunately, there is no widely accepted drug that can treat dengue or a vaccine to prevent it. Every year, 390 million people fall prey to this virus, and nearly half the world population is at risk. Normally, dengue is not that dangerous. Symptoms are mild rashes and the fever dies down after a few days. But in some cases, it can lead to the deadly dengue hemorrhagic fever or dengue shock syndrome, where the body loses water very rapidly and blood does not coagulate. The present treatment only includes close monitoring of fluids in the body. So our team is working on a strategy to target these PPIs used by the virus. We hope to design a drug that targets and breaks the interaction of two proteins in the dengue virus pathogenesis pathways as a possible intervention game plan. Small molecule drugs are organic compounds having low molecular weights and are often used to inhibit PPIs. But being small, they may not be able to cover the large size of the interacting surface of the two proteins, which are much bulkier. An alternative is larger protein-based drugs, which can effectively shield the interaction surface, but are not easy to deliver into cells by crossing the cell membranes. And so peptide inhibitors come to the rescue. These are drugs that are made of short sequences of amino acids, the building blocks for proteins. Taking the best of both worlds, these drugs are large enough to bind to the surface and can be taken up by cells. And this is what we are aiming for. Our PPI of interest is that between a viral protein and a host protein, which blocks a critical viral defense pathway in the hosts. This makes us unable to fight the infection, and the pesky virus happily divides and spreads through our body. To stop this interaction, we will screen peptide drugs and test how well they block the interaction. But how do we know that this drug will actually work in human cells? For this, we will express the two interacting proteins, viral and host, in human cells grown in the lab and make them interact in an experiment. Once the proteins are made in the cells, we will have to examine their activity. The method we'll use to observe our interaction is known as FRET, or fluorescence resonance energy transfer. We will tag these two proteins with fluorescent molecules, just like adding a label to find them among the cellular pool of proteins. When the proteins are far apart, we will see light of a particular color. This is because only one of the tags will be able to emit fluorescence. When the proteins come together, the fluorescent tag will transfer its energy to the tag on the other protein. When this happens, the color we observe changes. By noting this, we will understand how strong the interaction is and use this to test our drug. If the drug works, we expect the color to be the same as when the proteins are not interacting. And now you have a simple way to study the deadly dengue disease and design drugs.
Thank you for watching, and subscribe to our channel and follow us, the Team Berhampour and the Team Maastricht, on our social medias. Stay tuned for more amazing projects and great synthetic biology content.